Welcome to part 3 of my plant biology micro-lecture series. We'll be chatting about basic cell types today, but I'd like to preface this by saying this is not an exhaustive list. These 4 are just types that we'll see often and should have a good background knowledge of before moving on to plant tissues. So let's get started. Meristematic cells are the plant's version of stem cells. They are either entirely undifferentiated or only slightly differentiated, and as they differentiate, they become all other cell types found in the plant. You can usually spot meristematic cells due to their dark, prominent nucleus. As a side note, some sources categorize meristematic cells in their own group, while others classify them as Parenchyma cells, which we'll be looking at next. Before that, however, I'd like to give you a brief overview of the 3 cell types we'll look at next. These 3 types, Parenchyma, Collenchyma, and Sclerenchyma, are the real basic cell types that you'll see in most textbooks. I personally like to add meristematic cells into this group since I like to differentiate them from Parenchyma cells, but if you have a test question asking what the 3 basic cell types are, it will 100% of the time be Parenchyma, Collenchyma, and Sclerenchyma. With that said, let's move on to Parenchyma cells. Besides meristematic cells, remember they're sometimes categorized as Parenchyma anyway, these are the least differentiated of the group. They're also what we use in diagrams for a plant cell example, as they're the stereotypical plant cell with chloroplasts and a simple single cell wall. This graphic has great in-depth information, but the main takeaways are that Parenchyma cells have thin cell walls and have many intercellular spaces. Thinking back on our previous lectures, you should realize that the middle lamellas are not as prominent with Parenchyma cells. Lastly, that claim for filler cells is very accurate, as you find Parenchyma cells nearly everywhere, in leaves, flowers, stems, fleshy parts of fruit and fruits, etc. They perform a variety of functions, including storage, secretion, photosynthesis, transport, and response to wounding. This response may either be healing or adventitious organogenesis, which is just a long, scary phrase that means creating new roots or shoots in places you wouldn't normally find them. We'll talk about that extensively in an upcoming propagation microlecture. Our next type is Collenchyma, and I'm not a huge fan of how they drew these cells in the diagram. This is a cross-section of Collenchyma tissue, which is perfectly accurate, but the longitudinal point of view is much more telling, like in this image. If I lost you at cross-section and longitudinal, imagine cutting a Twizzler in half. Cross-section, longitudinal. I like the longitudinal image better since it actually shows you that the cells are elongated. Collenchyma cells also have thicker cell walls than Parenchyma cells, which usually get more and more uneven as they age, and they don't have chloroplasts. Their main function is to support young organs like stems and leaves. The strings in celery are a great example of Collenchyma, and they're the reason you tickle your tomato seedlings before transplanting them. If you haven't heard that yet, and may be a little creeped out, you either tickle or get a fan to blow over your tomato seedlings to replicate the wind they'll face outside. This disturbance indicates to the Collenchyma cells that they must thicken your cell walls to increase the strength and flexibility of these young stems before they get damaged in sudden, high winds once they're transplanted. Sclerenchyma cells can form from both Parenchyma and Collenchyma cells and are typically non-living at maturity. This means that the protoplast is gone and leaves behind 2 cell walls. That is, the original and primary cell wall, and then a secondary wall. The main feature of Sclerenchyma cells is the secondary cell wall, which has been lignified. Recall from part 1 of this series that lignin is a substance that greatly hardens the cell wall and renders it extremely rigid. There are 2 types of Sclerenchyma cells. The first types are sclerids, while the second types are fibers. Sclerids are referred to as stone cells, which protect other cells, found as the gritty bits in pears and around apple seeds, as well as the hard exterior of nuts and various stone fruit pits. Fibers are more elongated, much like Collenchyma cells, and provide support and strength for older organs, including bark. Hemp and flax are mostly made up of these fiber cells. After those 4, we're done. Next lecture, we'll be talking about how all of these cells work together to form tissues. Until next time, be sure to remember that meditation is for observing and accepting your thoughts, not stopping them completely. A common example is watching the sky and letting clouds pass by. You're not engaging with every cloud, just watching. Give it another try when you have the time, but do know that while it's incredibly helpful for some, it may not work for everyone. See you later.